So we'll start with the first talk, which is titled Unlocking Advanced Protein Manufacturing Using Systems Engineering. The speaker is Romel Menacho Melgar, CEO of Roke Biotechnologies. Romel earned his PhD in biomedical engineering from Duke University under the mentorship of Michael D. Lynch. He is the co-founder and chief scientific officer of Roke Biotechnologies, a Duke spinout focused on low-cost, scalable protein manufacturing. At Roke, he leads R&D manufacturing and commercialization efforts. His work focuses on developing standardized growth decoupled protein expression systems and engineering E. coli strains with enhanced expression and cell-programmed downstream simplification. Romel, all yours.
Thanks for that introduction, and thanks for inviting me here. I'm glad to be part of this. Let me share my screen to get this started. There we go. Okay, so before I tell everyone about Roke and our mission, which is to transform protein manufacturing, I want to briefly introduce our leadership team. So it's me, I'm the CSO and co-founder of Roke. I am the key inventor of Roke's IP portfolio. Then there's Guillaume Cotterell. He is the CEO. He joined Roke earlier this year. He has over 25 years of experience in biotech, and some of you may already know him. And then there's Michael Lynch. He is also a co-founder of Roke and advisor of Roke. He's a professor at Duke and a serial entrepreneur. So the problem that we're tackling is that traditional protein manufacturing is inefficient and expensive. So when you're trying to make a protein of interest, there's a lot of steps that go into making the protein. These steps are many times optimized in isolation. Then you have to integrate all of these parts into a final process, and that steps integration requires more optimization. Once you have arrived at a process that works and that is optimized for your protein of interest, this is specific to the protein that you're working with and at the scale that you're working with. So if you want to scale up, you have to return your process, or if you change the protein that you're working with, you have to return your process or even start over. Of course, there's additional problems such as pure protein expression, where the protein that you're trying to make just doesn't express well enough to support commercialization. And making the protein requires multiple steps, as I already mentioned. This requires a lot of expensive infrastructure, which is not only expensive to buy, but also to run. There's solutions too that tackle some of these, right? But again, they're context specific. So once you take one of these and you try to integrate it into or use it for your use case, that integration is non-trivial, and you need to do some adaptation cycles to get there. So instead of doing that, we have a systems engineering approach, right? And this enables a standardization. How? We have designed all the parts that go into our bioprocesses to work together from the get-go, right? So what this enables is process standardization. We have standard bioprocesses for the work for different scales for different proteins of interest. So you can scale up or down as needed, and you can change the protein you're working with using the same bioprocesses, and it will work. Perhaps more important than that is you can think about making advanced tools that address different problems within protein manufacturing. But if you design them to work in the context of our platform, then they become scalable and easily deployable to different applications. So we have solutions that increase expression, and we have solutions that simplify bioprocesses too. Well, what I mean by that is just reduce the number of purification steps to get your purified protein. So how do we do this? We use a two-stage autoinducible heterologous expression system that decouples growth from protein expression. What I mean by that is we let the cells grow until nutrient intermediate gets depleted. So once that nutrient intermediate gets depleted, your cells enter a stationary phase. They're no longer growing, they cannot grow. And then promoters in the cell become activated that drive the expression of your protein of interest. You can also co-express accessory proteins for different applications, or you can deplete host cell proteins during the production phase too. You can imagine, right, that if you change the protein that you're trying to make, the growth curve may look different, but you still induce a similar this when the phosphate gets depleted, and those conditions at which that happens are similar. So that's one of the reasons why we can get standard bioprocesses that work for different proteins of interest, right? This is already optimized. And something else that you get is because you are expressing your protein of interest when the cells are no longer growing, you're no longer limited by growth-associated boundary conditions. What I mean by that is you can deploy toxic strategies. For example, you can deplete essential proteins, right? Because your cells are no longer growing. So one of the first things that we did to simplify manufacturing, right, we wanted to create an autolysis strain. So we have standard bioprocesses. Can we make a standard autolysis strain that can be lysed without the use of equipment for high throughput applications? So the way we did this is we co-express in the production phase a lysozyme and a nucleus. The nucleus goes in the periplasm, so it cannot come into contact with the host cell DNA of E. coli, so the cell doesn't die. And then the cytoplasm is the lysozyme is in the cytoplasm. The cells are stable at the end of fermentation, but if you push them a little bit with a freeze-thaw cycle in the presence of detergent or heat, you trigger cell lysis. The lysozyme degrades the cell membrane, and the nucleus can go into the cytoplasm and degrade the whole cell oligonucleotides. These are the results from that. As you can see, compared to a control strain, right, our autolysis strain results in virtually complete protein release. This is compared 100% is sonicated samples with both triggers, right? And because you're getting rid of the host oligonucleotides, you're getting non-viscous lysates, which are ideal for high throughput applications. You can imagine that you can grow and express your cells in multiple microtiter plates, and you can freeze thaw them at the same time and lyse a bunch of plates at the same time without needing to use, for example, a 96 volt plate sonicator. But another cool application of our autolysis strain is because you can elicit the lysate with heat, you can do this in the bioreactor, right? So we have this thing that we're calling in-bioreactor purification, where we express our protein and we purify it. This works for thermostable proteins in the bioreactor, right? So you basically heat up the tank, the cell lysis happens, you add some precipitation agent, and then you crush everything out except your protein of interest. And in one step, right, we get pretty remarkable results for multiple proteins. This is just a couple of examples for molecular biology enzymes. And you can see this is an SDS page of that purified protein coming out of the bioreactor. And this is for a pharma grade protein, right? Because we can make this is a very simple manufacturing process. You don't have oligonucleotides too. We can make this for really cheap. Our portfolio we're selling this in our website if you're interested in what our portfolio looks like, and it's always we're trying to launch more enzymes in the next few weeks. But also, I think it's important to note that we've done this with a pharma grade protein, right? Because this was for pharma applications, we looked at this using mass spec, right? And we couldn't find anything else. So this is 100% pure by mass spec standards for this protein. I'm not saying that for molecular biology enzymes. That is to say that we can get it to work really well. So you can imagine that this simplifies manufacturing, right? But I also mentioned that we have technology to express hard to express proteins. So we made a strain that is able to express nanobodies. Nanobodies are single domain antibody fragments that require disulfide bonds for proper folding. This is not, you cannot get disulfide bonds in the cytoplasm of E. coli because of reductases in the cytoplasm, and you can't really knock them out directly because the cell will die. You can knock them out and have some compensatory mutations, but then you don't have a robust growth. But in our case, we can get a wild type growth with the reductases being there during growth. But once we go into stationary phase, we get rid of them, right? So what we're basically changing the phenotype dynamically of the cell to something that it looks the wild type to something that is optimized for the expression and the formation of the disulfide bonds for nanobodies. So these are our results from an antibody panel. In dashed lines here, you can see the highest reported titer for nanobodies in an E. coli system, both for cytoplasmic system and a periplasmic expression system. And you can see that using our E. coli strain, we surpass those previous benchmarks across the nanobody panel, right? It's important to say that we're not retuning our bioprocesses. This is the same bioprocess that works for over 80% of the time. So this is pretty remarkable. On top of that, I mentioned, right, we have scalable bioprocesses, so we can do this in microdiator plates, shake flasks, or bioreactors. And on top of that, we can combine our tools as we want to, right? So we have put our autolysis strain into this BHH strain, this nanobody strain. And the result of this really is a platform that allows for some rapid prototyping at small scale, microdiator plates. And then once you have identified the lead, you can rapidly scale it up, right? We have obtained up to 6 grams per liter intact and medium cell density. So in theory, we could triple this number. If I have to summarize, we use a systems engineering approach that enables the development of scalable and easily deployable manufacturing solutions that increase expression or reduce cost. If you want to contact us to explore collaborations, licensing opportunities, or partnerships, you can email us at support@roke.bio, or you can use our contact form in our website. And with that, I'll take any questions.
You talked about a great talk, by the way. Thank you. You talked about cost reduction. I may have missed the detail. Which part contributes most to the cost reduction in the overall process?
Yeah, so we've done. It depends on, obviously it depends on what process you're working with, right? But we did some TEA analysis on the precipitation-based purification for that pharma molecule that I talked to you about, and we get over 50% cost reductions on both capital expenses and cost of goods sold. And the reason why you get that is because you get rid of chromatography steps, right? So the only thing you do is you have your tank, you heat it up, you add some precipitation agent, you get rid of everything except your supernatant, and then you can do some endotoxic removal and glucose formulation, right? So it basically eliminates 2 chromatography steps and the diafiltration steps that are associated with getting those in the buffers that are required to do those chromatography steps.
Any question? There we are.
Hi, great talk and interesting system. In the case of the VHH, the nanobodies that you're working with, have you done any functional tests to show that your purified protein is not only pure, it's actually clinically relevant?
Yeah, thanks. So yes, we have. So they are properly folded. We've even done dimers of nanobodies, and they express well too and work well.
Thanks. If no further questions, we can thank the speaker once again.
Thanks for having me.
Thank you.